So it is 3 minutes past the hour, and I think we will get started. And I've seen all our speakers are here already, which is the most important prerequisite for this to work. And we are very happy to present to you our new set of ILTER research initiatives. And of course, there's no nicer way of introducing this than using the nice flyer which Deshani has created, where you see all our speakers. And you can also see that we have a wide variety of different topics and also different countries where people come from. So from around the globe, we're very international here, very diverse, very mixed topic-wise, geography-wise, and so on. So we are very happy about this outcome. And as Ellie has already pointed out in his report of the Science Committee, we have received 80 applications. Out of these, we have selected 5 through a quite vigorous selection process. So this is, first of all, of course, scientific excellence, but we also have other aspects there. We like to have these initiatives to have an educational aspect. Some of these are part of thesis work. And it's also important that they foster the networking aspect. So all of them work across different sites because ILTER is a network, and it has, of course, an added value by combining the work of single sites. So it's more than only the addition of them. And we also want to have some diversity in topics. So we have a set of criteria. You can see this on the website. The website address is there. The link to the page with these research initiatives, and only a short word of what these research initiatives are. We have revised this concept in 2023 completely, so that we now invite these through a call. So it's a call on our website, which is published internationally, and so it's also spread through our different communication channels by the different networks involved and also our own channels. So we publicize it widely so that we really get applications from all of the different ILTER networks. And this is actually a very nice way to spend ILTER funds because we are funding here some active research. And especially since we are also supporting early career researchers, I think this is a very good investment in the future of the ILTER network. And we're happy, and I'm actually also a bit proud that we have such a great outcome, and I very much look forward for this seminar. And with this, I will hand over to Deshani, unless Ellie, you want to add anything. Apparently not. So then, Deshani, I hand over to you, and you will guide us through the program.
Thank you, Herbert. Our first speaker is Veronica Nava from University of Milano-Bicocca, Italy. Veronica's project title is Freshwater Innovations in Research and Education, an Italy-United States partnership. Veronica, please.
Yeah, am I supposed to share the screen, or will you share the screen for the presentation?
You can share the screen. If you want me to
Okay.
share the screen, I also have your presentation loaded.
No, it's okay. I have it. Thank you so much. Can you see it?
No.
Herbert, could you stop sharing your screen?
I can stop sharing here.
I don't know. Do you mind sharing it? Okay.
Yeah, I will.
Okay. Thank you. Thank you.
Okay.
Okay. Thank you. Sorry for this. So good morning, everyone, and thanks very much for organizing this very interesting webinar. As Dishan said, I am Veronica Nava and currently a postdoctoral researcher at the University of Milano-Bicocca in Italy. And in this presentation, we talk about an expedition that we have performed on different lakes to collect high-frequency, spatially explicit data to assess water quality and gas emission in lakes. Next slide. So the focus of this presentation is lakes. So we know that lakes are a fundamental ecosystem, and what is interesting about lakes is that they occupy a relatively small area globally. But despite this, they support a remarkable biodiversity of species, and they provide a huge variety of ecosystem services. And this goes from raw water supplies to food, hydropower, climate regulation, and also all the recreational and aesthetic values that this ecosystem holds. Next slide. So there is a long tradition of limnological research, and with this tradition, we have collected many long-term data sets on lakes. And these are fundamentally important because we understand how these lakes are changing for climate change and different anthropogenic pressures. But often, all these data have been collected just in one point. So lakes have been mainly conceptualized in their vertical dimension, and we do not know much about how lakes vary among the spatial dimension. Next, please. Also, the majority of this research has focused on the open water zone, so on the pelagic zone. But lakes are embedded in the terrestrial landscape, and they can be profoundly affected by the surroundings. For instance, inflowing rivers are so important in determining how the lakes can change. And these effects and effects also of human pressure on lakes can be mainly seen on the littoral zones that are really the nexus of interaction between humans and lakes. But as I said, not much of the research has been done on these littoral areas, and much of the research has been done on the open waters. So we're kind of not knowing much what is going on the littoral. Next, please. Among the different water quality parameters that we can observe on the spatial scale in lakes, we can also have greenhouse gases, because we know that lakes also are significant sources of both CO2 and methane. And so it would be interesting just to understand a little bit more about how this methane can be produced also from deeper and larger lakes. Next, please. So given this framework, the aims of this project was to start quantifying the role of lakes in greenhouse gas emissions and considering the spatial heterogeneity in lakes. So we want you to understand if we have a contrast or how we can actually harmonize data that have been collected in a single point together with spatial distributed data. And we are especially interested in seeing contrast between pelagic and littoral areas, and also looking into the effect of inflowing rivers. And also another aim was to enhance the integration of different monitoring data sources on lakes to better comprehend how these lakes are changing and how they function, including the long-term data set with high-frequency single-point sensors and those other technologies like remote sensing. Next, please. So to do so, we have developed this project in which we have a partnership with the University of Nevada-Reno in the United States, and we have selected 3 different lakes: Lake Tahoe in the United States, Lake Iseo, and Lake Maggiore, located in the northern part of Italy. All of these lakes are deep lakes and large lakes. They're very important for their economies, and for all of these, we have long-term data set that we can analyze and combine with the picture that we can obtain. Next, please. So what we have used for the project was this flame technology. So this was developed by the Center of Limnology, University of Wisconsin, Madison. And this is basically a platform that allows to collect many different parameters all together. So it has an intake manifold that goes into many different sensors. So while you're cruising along the different lakes, you can gather real-time data and special species data. Next, please. And specifically, the parameters that we have measured with the flame technology are temperature, electrical conductivity, pH, the fluorescence component of the solid organic matter, chlorophyll A, and also phycocyanin, so blue-green algae, nitrates, methane, CO2, and nitrous oxide. And together with this, you can also have information about your basin, like the GPS, together with the bathymetry of the lake and so on. Next, please. So far, what we have achieved was to perform this lake expedition on 2 out of the 3 lakes that are included in the project, so the 2 Italian lakes. And we have done this expedition the last summer. We have 13 different people involved, and we have students, early career researcher, professor working together. They were coming from 4 different institutes. So we're starting setting up all the instruments and test them on a smaller and shallower lake, because as you can see in these photos, there is a lot of electrical gears and connection that needed to be done. Next, please. And after these, next, please, sorry. So, and after this, we started our actual sampling on Lake Iseo and Lake Maggiore. Sorry, the previous one, sorry. So we actually navigate for around 50 hours along the different lakes. We have collected more than 3 million single points on these lakes. And besides these, we have also collected some other parameters to combine our long-term data set with these. So we have also collected some samples from the vertical dimension. So the process in which we are now is the data elaboration phase. So we are cleaning all the data. So we need to remove and check the data, remove up layers and anomalous values. We have to apply some temporal correction to the data to consider the sensor response. So the time that elapsed between the moment in which you collect the samples and when the sensor responds. And we have created the first map, this rough map that you can see here, which are basically the transect that we have done on the different lakes. But these still need some elaboration. Next, please. And so it's just the last slide. So and that indeed, so yeah, we just need to, the next step will be the collection of the samples on Lake Tahoe and to really interpolate all the different maps. So just the next one, I want to acknowledge all the people that were involved in the project. And with this, I've concluded that I'm happy to answer to any question that you might have. Thanks.
We'll move on to the next presentation and we'll ask questions at the end if time permits. Our next research initiative is titled Long-Term Ibero-American Study on Nitrogen Deposition. The presenter is Madriaza Maturana from Institute of Ecology and Biodiversity, Santiago, Chile.
Hi, everyone. Can you see my?
Yes, we can see your presentation.
Okay. So, hi, everyone. Good morning. My name is Karina Madriaza, and I am postdoctoral researcher of Institute of Ecology and Biodiversity of Chile. And today I'm going to talk about Liaison project. Some key aspects to consider for my proposal. Ecosystems are being rapidly altered by factors such as rising carbon dioxide levels, habitat fragmentation, and human activities that disrupt the global nitrogen cycle, including fertilizer production and fossil fuel combustion. The nitrogen cycle is unique due to its large atmospheric reservoir of nitrogen gas and the biological mediated conversion of nitrogen into chemical form usable by most organisms, with nitrogen circulating between plants, animals, microorganisms, and the environment. Human activities has doubled the transfer of nitrogen from its atmospheric reservoir to biological available forms, increasing nitrogen fixation and leading to higher emission and deposition of nitrogen gases like nitrous oxide, nitric oxide, and ammonia. Human alterations to the nitrogen cycle have led to increased nitrogen input, higher greenhouse gas emissions, soil nutrient loss, acidification of ecosystem, greater nitrogen runoff into water bodies, increased carbon storage, accelerated biodiversity loss, particularly of nitrogen efficient species. Next. Here. So why is relevant to measure and quantify this? In southern Chile, nitrogen deposition is understudied, but the region's ecosystem influenced by maritime precipitation may represent some of the closest conditions to pre-industrial level globally, offering a unique, pristine environment free from pollution. Why conduct a nitrogen deposition study in the long-term socio-ecological research Chile network? Studying nitrogen deposition in Chile's pristine forest is crucial to understanding global environmental changes. This research aims to measure inorganic nitrogen concentration across a latitudinal transect from Atacama to Magallanes, providing valuable data on nitrogen deposition and its impacts on soil pH in Chile's protected ecosystem. So, why is my research question? How does the health of ecosystem in a latitudinal transect respond to temporal variation in ammonia and nitrite in rainwater? So, work will be carried out at the 9th long-term study sites in Chile of the long-term socio-ecological research network, which are influenced by different climatic conditions. In this way, study will cover Chile latitudinally, including sites from the Tarapacá regions like Atacama Station to Magallanes and Chilean Antarctica region like Omora Park. Work will be carried out at the same time at 2 sites in Colombia and at 2 sites in Argentina that have the potential of long-term study sites. We are proposing to establish a long-term monitoring network across 29 sites in different ecosystems in America, 13 in South America, and 16 in Europe, Spain. This network aims to quantify the deposition of nitrogen and its potential effects in the world. So we will collect water samples from the field, in the rainfall in the field, in the lab, we will prepare the ion exchange resin and we will obtain the ions of the solutions. And then we will measure the ion concentrations using spectrophotometry. We will also record changes in soil pH at each study site. And how is nitrogen deposition measured? Currently, the most efficient way to measure the concentration of ammonium and nitrate in rainfall is by using mixed bed anion and cation exchange resins such as amberlite, a procedure that takes advantage of the properties of the resins capture and retain the ions present in rainwater. So what we do expect in this project? First, to provide biennial measurements of inorganic nitrogen concentration in precipitation and capture water at long-term studying sites in Colombia, Chile, and Argentina. Second, determine change in soil pH and water qualities from nitrogen depositions, and at the same site. And third, to compare the collected data with the obtained from developing a monitoring program of air quality and effect in the Spanish National Park networks research, which use the same methodology and the temporal context. By having measurements of ammonium and nitrate present in the rainwater in each of the sites, I will be able to estimate first, how much long nitrogen enters and leaves to the ecosystem, where it is stored or where it is lost. And I will be able to determine which of this ecosystem is most affected by the deposit, generating a risk map through monitoring water quality and soil pH, understanding the sensitivity of ecosystem to the modifications caused in nitrogen cycles. It's important to me to clarify that this project has not yet started. The first steps involves a trip to Spain to the CEMAT Center for Energy, Environmental and Technological Research, where Dr. Rocío Alonso will teach me the necessary lab techniques to carry out the methodology. This trip is planned for the end of this month. After that, I will share what I have learned with two students, one from Colombia, one from Argentina, who will travel to Chile for this purpose. Once they return to their respective countries, we will begin collecting data in parallel. Once the data collection is completed, they will send me their samples, which I will process at the lab of Catholic University of Chile, where I work. Finally, we will compare this result obtained, those collected simultaneously by the research group led by Dr. Rocío Alonso. Thank you.
Thank you, Madriaza. Next up, we have Merav Cohen joining us under challenging conditions from Israel. Merav's presentation title is COVID-19 as a Major Global Disruption to Examine Social-Ecological Systems Resilience Theory. Thank you for joining, Merav. We understand that if you have to leave the presentation as you have informed us.
Thanks so much for organizing, Deshani, and thanks for inviting me. Yes, conditions here in Israel have been difficult. I hope that we can go on without interruptions. Let me just share my screen. Let me know if you see this okay.
Yes, we can see your presentation.
Perfect. Well, hello, everybody. My name is Merav Cohen, and I study what makes social ecological systems more resilient. This is going to be a very sharp turn away into qualitative research. So those of you who are interested and want to learn more, then this is the time to pay attention. It's going to be very different from what we've heard so far. And thanks for the other presenters for all this really impressive work that they've presented. So the main research question is, what social ecological attributes can strengthen or weaken a society's resilience in the face of a major unknown and unpredictable crisis? Before diving in, though, let's just make sure we understand resilience in the same way, because it is very much a slippery concept used differently in various fields. Our working definition is that a social ecological system is resilient if it has the capacity to cope with disturbances without losing most of its existing defining functionality. Now, resilience is often studied with regard to a specific place, time, and disturbance. For example, the resilience of rice fields in Thailand for flooding, or the resilience of indigenous populations in Alaska to climate change, with regard to a particular time in which it's estimated. But there's a growing body of research that aims to generalize things so that we can be better prepared for the unknown, meaning to identify attributes or qualities that are to be associated with the coping capacity of a social ecological system. And these principles can refer both to the structure of the composition of the system, like diversity of its components, or connectivity levels between them, finding that sweet spot. It also can relate to stores of its components. Does it have reserves? Does it have social capital, for example? It can also relate to competencies of the system to carry out particular activities like learning and innovation. And the examples here are principles which have been most supported by the literature according to a review that I've conducted. And these are the focus of my work. And COVID-19 we identify as an opportunity to address challenges in testing and comparing these principles of resilience across different social ecological systems. Systems that vary in their structures, their mechanisms, and external conditions, all we're facing the same disturbance practically simultaneously. And this is the big opportunity here. So essentially, we're looking at the system holistically and focusing on the societal impacts from COVID and societal resilience, drawing from these principles of social ecological resilience. So essentially asking to see whether these principles played a role in building or weakening resilience to the impacts of COVID, and if so, how? And to do this, we designed a comparative study of 3 diverse social ecological systems, each a defined platform for long-term socio-ecological research, LTSCR platforms, which are a part of both ILTER, the European, and the ILTER, the international networks, all in collaboration with local research teams. We conducted interviews on the ground, aggregated their results, collected background data, and then fed that whole thing back to a group discussion. More specifically, if you're interested in some qualitative methods, we produced causal loop diagrams from each and every one of these interviews. We created them with the stakeholders to identify how they perceive the impacts of COVID, how they may be connected, and what may be the drivers or the mitigating factors for them. We used Mental Modeler to do that. Results from each participant looked very differently. I'm just flicking through 2 examples from Taiwan here, from the Social-Ecological research platform in Changhua in Taiwan. And you can see how this may be challenging to aggregate, just to kind of give you a taste of what this looks like. So in aggregating the results, clustering them and aggregating, things got fuzzy. If you're not familiar with fuzzy cognitive mapping, it's essentially causal loop diagrams that we put weights on the different parts of the map. So here, we created a fuzzy map that in which the weights convey the level of agreement by participants, both with regard to each node that was identified and the links between them. So we have this core of high consensus all the way out to a fringe of things that were only mentioned by a mere few. And this is what it looks like in Taiwan, just to give you a taste. So you have the high consensus in green, the medium level in yellow, and we excluded the red just to keep this manageable for conversation so that we can have stakeholder feedback. In addition to the aggregated map, we also collated data regarding the impacts of COVID, both direct and indirect, such as mortality and unemployment, and with regard to the characteristics of the place, like demographics, the sectors that operate there, population density, and then circled that back to the same stakeholders we interviewed to reflect both on our analysis of their experience and these official numbers, with the idea to generate a lively discussion to deepen our understanding of these narratives. Just as important, however, was the mere interaction among the stakeholders. So really walking the talk of transformative social ecological research, sharing the knowledge, building relationships, as well as the capacities of the participants, of these stakeholders, to think in systems. We are now in the crucial stage of analysis, where we ask, how do these principles emerge from the collected narratives? And how do they correspond with COVID-19 impacts in the region, both experienced and from the official data that we've collected? And here's an example of how we're thinking of this. This is the money time from the Taiwan study again. These are our initial observations following the network analysis of the fuzzy cognitive map, where we identified key impacts, drivers, central nodes, and receiving nodes. So here's one example. If we're looking at diversity, all right, for example, this is highlighted through the strong impact that COVID inflicted on tourism, really a fundamental pillar of the local economy. And finding that optimal connectivity, to be connected but not too connected among the components of the system, is reflected in the characteristic of the local population, which is in the driving factors of low density on the one hand, and on the other hand, very close social ties. It also surfaces through the driving role of the media as a connector during COVID, if you recall, and the central node and the central role of reducing personal contact among the people. The emphasis on close community ties as a supporting mechanism against these aggravating factor of minimizing personal contact also indicates a principle of social capital. This is here at the third one. Having sufficient amount of vaccines indicates having reserves, either the stock of the vaccine or funds to purchase it, or the capacity to manufacture it. And the ability to adjust our business models to deal with the new reality, that shows learning and innovation capabilities. So what would this look like when we compare across 3 sites? Stay tuned for that. I hope to be able to share that with you in the future. But mostly I hope that this may inspire you to take advantage of this comparative possibilities, opportunities that these networks provide, because my experience has been that really, while this research doesn't use long-term data series, it's our affiliation with these networks that enabled collaboration with the local research teams, building on their established infrastructure, their local knowledge, their long-time relationships with stakeholders. And this enabled us to find and access data and translate and contextualize this work for each of the locations. If this is interesting to you, I welcome you being in touch because I'm scouting for another couple of locations and a postdoc opportunity to make it happen. So thanks for listening.
Thank you, Merav. Next up, we have Baptiste Wijas from University of Miami, United States. Baptiste is going to talk to us about interactions between drivers of wood decay and their contributions to carbon cycling.
Thank you, Deshan. So today, I'll be talking about deadwood in ecosystems, various ecosystems, basically, and how it relates to carbon cycling. So I'm not at the University of Miami anymore. I'm at the Kerr Institute of Ecosystem Studies and at the University of Queensland. I'm at 2 affiliations now. So deadwood carbon stocks, which is basically when a tree dies, it basically has a lot of dead wood that's left over once it dies. And so these represent about 8 to 10% of global forest carbon stocks. And so deadwood stocks are often assumed to be these stocks that are quite static through time, because deadwood is quite recalcitrant and kind of thought as this thing that's always there in ecosystems. But they can actually fluctuate quite dramatically. For instance, in this picture, we can see here what happens after a windstorm, where you get this huge influx at once of deadwood into the ecosystem, which can have consequences for how we understand how it decays, basically. But at the moment, we have quite a small understanding of how these deadwood carbon stocks fluctuate through time, as most surveys are carried at one time point. And so deadwood decay is carried out by many different drivers. So deadwood decay is essentially what needs to happen so that deadwood is transferred either to the atmosphere here as CO2, or then transferred up into food webs, depending on what eats the wood, or it can go back into the soil as soil carbon. And so there's all these different drivers, whether abiotic, so we've got fire, sunlight, water, freeze and thaw cycles, which kind of drive how the deadwood decays. And then we've also got the biotic factors, which actually consume that wood. So we've got the fungi, which are pretty common in consuming wood, termites, or even humans by actually harvesting wood. And so we kind of know how each different component kind of decays wood on its own, but we don't really know what happens with all the interactions between all of these different factors and how they come and interplay together to ultimately influence how deadwood actually decays and how that relates to CO2 emissions. And so research in the past, I'd say maybe 5 years, 3 to 4 years now, has really gone quite deep into looking at the interactions between different biotic drivers of deadwood decay. So as we can see, there was this Nature article about 3 years ago looking at the contribution of insects, and there was a Science article looking at the influence of termites and the interaction with microbes. There was a paper in 2019. So many different papers have really started to look at these interactions between these biotic drivers. But what we don't know is what happens with the abiotic drivers and how these interact with the biotic drivers themselves. So the abiotic drivers I'll be looking at in this project are both fire and water, so rainfall. And so fire in itself basically chars the wood. Depending on the intensity of the fire, you can still have a lot of wood left behind, but that wood will be chemically modified compared to what it was before the fire came through. And then water also can modify wood by essentially allowing leaching of nutrients, which can have consequences for how the nitrogen and carbon content of the wood. And so together, we don't really know how these influence how different biotic drivers of decays, whether it's fungi or termites, will actually decay that wood that's been modified by this fire and this rainfall. And so I've got 3 main research questions for this project, which are, what are the legacy effects of abiotically modified wood on biotic-driven wood decay? Then we're interested in whether there are ecosystem differences in how these biotic drivers interact with these abiotic drivers, depending if fire is quite prevalent in the systems or depending on whether there's a lot of rainfall or not. So trying to understand these interactions, whether they're context dependent. And finally, we're interested in looking at whether there's temporal variation in deadwood carbon stocks and what their actual contributions are to ecosystem fluxes, basically. And so my experimental design is built in 3 ways. So first, I'll be running a wood decay experiment. Second, we'll also be doing deadwood surveys and termite surveys. And finally, we're kind of going to put it all together to model the contribution of deadwood to carbon cycling across different ecosystems. And so wood decomposition experiment, basically what's usually done is that you set out wood blocks for a given period of time. So you set out these wood blocks on the ground in the floor, you measure the mass at the start, and then you measure the mass when you collect, and this gives you an indication of how much mass loss happened during that time, and so how much carbon is being lost from that wood. And so I'll be setting up that experiment across 6 ILTER turn sites, so the Terrestrial Ecosystem Research Network in Australia. And so these sites range from cold alpine sites down here to basically tropical savanna rainforest sites up there. So we'll get a real gradient in temperature and rainfall. But this time, so this kind of experiment has been done a fair few times before. This is not very novel, but we're going to put a bit of a twist to this experiment as well, where we want to really look at that burnt and water leached wood. So basically, we'll set out some slightly charred wood out in the field. And so I've been starting to char the wood before putting it out. So using these fancy machines that can control how wood is burnt. And so I can essentially create a charring layer on top of the wood, and then I'll set these out to see how the burning actually affects decomposition. And then for water, I'll essentially soak the wood blocks a few days and then take them out and then put them in the field, and then compare if there's differences in how wood decays, whether it's been charged, water leached or not. And so the other component of this project is to do deadwood surveys. And so about 10 years ago, there's been first some deadwood surveys in all of the sites I've been to. So I'll be doing a second collection of deadwood surveys, which will start creating a long-term data set of how deadwood stocks have actually changed during that time. And I've also partnered up with a private company which is specialized in LiDAR. So basically LiDAR is a method used to measure carbon stocks in forests using laser scanning and some kind of remote sensing method, basically. And so I partnered with Ibero-American, which will help us to really try to see if we can map deadwood carbon stocks using LiDAR technology, essentially. At the moment, LiDAR technology is really used to map above ground biomass, but we don't really know whether it can be used to map deadwood stocks. But here's a first example of a site we scanned about a week ago, which shows all the deadwood mapped using lasers, which is actually quite cool. And so then we'll also be doing termite surveys at all the sites, which will add to the TERN data sets. And so to try and relate termite community composition to decomposition rates. And so finally, the aim is to kind of put this all together. So at all of these TERN sites, there's carbon flux towers, which have been operating for 5 to 10 years. And so we'll get a measurement of how much carbon is emanating from these ecosystems, then knowing how much deadwood is in these ecosystems, how much has changed, and then also knowing how, from the wood decomposition experiment, the rate at which this wood decays, we can kind of hopefully combine it all together to really get a good picture of how deadwood decays across all these systems and how that actually contributes to the deadwood carbon cycle. And so hopefully we'll really try to improve the estimates of this contribution of deadwood through time and through space, which can help researchers around the world to try and understand carbon emissions a bit better emanating from this big stock of carbon. And so that's me for now. Thank you.
Thank you, Baptiste. Our final speaker for the day with an equally interesting and exciting project is Pablo Mendez, joining us from Institute of Innovation and Knowledge Management from Spain. Pablo's project title is Averting Biosphere Lock-ins in the role of standardized cross-scale long-term socio-ecological and biophysical research.
Thank you, Dishani. Thanks for the organization of this amazing webinar. Thanks to my fellow presenters for their great presentations. I'm glad that Merav opened the door to qualitative research. I'm going to give you another dose, but hopefully you will see that we are trying now to quantify the phenomena that we are investigating in this research. I'm Pablo Mendez. I'm from this institute from the Spanish Research Council. I've been collaborating with ILTER for many years now, and this is part of this ongoing collaboration with LTSER platforms over the last year and a half. The scope and the main questions. I'm going to move my Zoom here. The scope is how to understand how the combining impact of different environmental change effects over the erosion of ecosystem functions that are providing services to upon which local and global communities depend. And this erosion is increasing the likelihood of lock-ins at the system level, which are situations where key system components are strongly intertwining and make the system resistant to change towards alternatives, whether more or less sustainable, but we're interested, of course, in sustainability, sustainable initiatives and solutions. These dynamics pose high risk of irreversibility, that we reach to a point where we cannot reverse our effects on these critical ecosystem functions and other key services. So our question is, how do these local mechanisms affecting these risks interact across local to global scales, preventing the sustainable outcomes, and a key question is that we don't know is are these contributing to in a cumulative way to state shifts at the biosphere level? And what is the role of long-term standardized observations to understand and prevent systemic risks at this level? They are not going to stop too much here, but the basic, the conceptual background again is resilience theory, and in this case, the concept of traps. The questions here, cutting across all these trap types, is what is the level of rigidity and risk for locking in a system is, and we conceive like this is to be in a continuum, so poverty, rigidity, and locking. And one of the key factors scarcely explored is the potential of social-ecological change in sustainability transformations. Another conceptual, another important concept is this concept of pathways of development. We are situated here. You can see my cursor on the red dot, which is between history and the future. The outcomes in social-ecological system emerge from relationships among different factors, like resources and institutions, the actors and the strategic situations in which they find themselves, power dynamics in governance, and is of course context sensitive, so sensitive to a local scale. And we have the problem of directionality. What is a sustainable pathway considering, multiplicity of values, beliefs, and interests? And relevant here, of course, is this path dependency. So once we take a path, there are one regime among different systems selected, and this can be locked in and can lead to reversibility. So important here also is that history aggregates past decision events, environmental change and interaction, but also carries variation, carries diversity, heterogeneity resulting from legacies. So there is this tension between history and opportunity for the future. And in this tension, what is the opportunity for change through adaptation and transformation? But locking is generally seen as a negative phenomena characterized by minimal opportunity and potential for change towards alternative development pathways. So the research that we'll do through this ILTER funding is developing a method, but it's based on past research, collaborating with experts and actors from the 6 research LTSER platforms in Europe, where we synthesized a testable conceptualization of looking as a boundary object or a bridging concept. And we distill 5 necessary conditions to derive observable components of system locking, which are ecological suppressions versus ecological restoration. So what is the link and the tension between ecosystem functions and socioeconomic processes? They are linked. The set of beliefs operating within a governance setting. So do we believe in applying techno-scientific solutions or working with nature? Pluralism, what is the diversity of epistemologies and ways of knowing at the local level? The degree of centralization of decision-making, and this is also very much related to power asymmetries within the governance system. And what are the costs for transformation? And the concept is here shown by this ball and cap metaphor, which is very much used in resilience theory. Here you see the necessary conditions. And so depending on this combination of these observable components, we can reach sustainability measures or impacts like ecological suppression that can deepen locking and increase the risk of irreversibility. And as I said, very important is the relationship between what is happening at the micro level, how are these telecoupled with other social-ecological systems in distance, like an agri-food value chains, for example, and what are the links with and the effects at the biosphere level. So I have no method to show because this ILTER funding is to develop a method collaboratively with platforms, experts, and managers to develop a mixed methods. So combining qualitative research with quantitative research, to utilize long-term standardized observations to understand the cross-scale social and biophysical causal mechanisms underlying these lock-ins, and identify the spatio-temporal scales at which lock-ins can operate and how they could propagate across scales, inducing global systemic risks. And for that, we are using 8 case studies across ILTER platforms, 4 for developing the methodology and 4 for validation. And we are starting now the process for communicating and involving the platforms. And we will use another project because this is a broader design within the soil mission, living labs for reflection purposes. So we will use the methodology to reflect about lock-ins in the agri-food sector in these cases. So thank you very much for your attention.
Thank you so much, Pablo. We have about 5 minutes left for us for a discussion. If you have any questions, you can directly ask from the speakers now. I see that there have been a few questions in the chat and being answered. Merav, you have one question pending. Would you like to take the time to answer that?
Sure. Hi, thank you. Thanks for the questions. So I'll start with the easy question by Jan. Jan asked, how did the stakeholders respond when we showed them, when we reflected back what we found? So just to recap, we aggregated the responses into a fuzzy cognitive map of how they see the experience of COVID-19, the different components and the interactions between them. And then as you would imagine, the things that were in high consensus, these went pretty quickly. The red or the components that were more in the fringe that were only mentioned by a few of the people generated a very lively discussion, and it was super interesting. The experiences, so we've completed this in 2 out of the 3 locations so far, in France and in Taiwan, and the experience was very different. And I think that this is because the experience during COVID was different, and the relationship with the government is different, how information was shared and how information is dealt with in each of these locations as well. So you show publicly available data, official data, and I can tell you that the responses were very different. In one of the locations, people were like, yeah, that makes sense. And then the other was like, no, that's a lie. And I'll keep you on the edge to learn which one's which. It's sensitive. It was very sensitive. But I can tell you that just breaking the information down into the level of agreements really enabled us to focus on where there was contingencies and to fill in the gaps really well. So I'm really glad we did it in that way. The second question by Cremona, do I have time, or should I respond to Cremona offline, or are there other questions?
Let's see if the audience has any other questions for the other speakers, and if not, we can move on to the second question. Okay, you can go ahead.
It's very wordy. I was trying to listen to and didn't want to take time to write this down. So Cremona asked about the definition of resilience. So if you recall, I said that our working definition coming from the field of social-ecological systems research is that a social-ecological system is resilient if it can cope with disturbances and maintain its defining functionalities, right? So obviously, I didn't have time to go deeply into it, but in this theory, Cremona, that there is the underlying inescapable premise that we're geared to maximize human well-being. It's an anthropocentric approach. So, you know, resilience isn't good or bad. And Pablo touched on that a little bit in his talk as well. Resilience as lock-in, neither necessarily good or bad by definition. You can think of many situations that are resilient, but are undesirable, like a totalitarian regime, for example, right? Or even within the ecosystem, you can think of situations that are very resilient, and these are these lock-ins. But when we're looking at the defining characteristics of the system, then we're looking into how society is still society as we know it. And when you bound your system, when you define the system that you research, right? Are we looking at a city? Are we looking at the country? Are we looking at humanity at large? You need to make that call. What are the defining characteristics of the system? And at what point do we tip over and the society that we research is no longer the same society? So you can look at the society in Taiwan and that small coastal community that is mostly based on agriculture, and it has a particular societal structure. And you need to define at what point does a shift in the conditions of that socio-ecological system makes us something else. It's no longer a forest, it's now a savanna, right? So think about that in terms of the social system. So it's an excellent question, and one that we have to tackle right at the beginning of the study.
Thank you, Merav. Thank you so much, all the speakers. One last thing I want to say before I hand it over to Herbert. Esteban Arrieta, International Management Office's undergraduate intern, had been a great help organizing this webinar. A big shout out to his help as well. Herbert, would you like to take over to end the webinar?
Yeah, we can do that. There's not much more to say. Thank you so much to Deshane for arranging, for Esteban for helping, and of course, especially to our speakers. I think this was one of the best webinars we ever had. This was really nice. I really enjoyed each and every talk. I'm really grateful. And it's also nice to see you kind of in this official way, because all of us have been in contact more on the administrative level, right? Sending plane tickets and restaurant bills and so on. So it's also really nice to now have this, this scientific output, and so the real purpose of the whole thing. And of course, I'm also familiar with your proposals, so it's really nice to see how this work is evolving, and we are all looking forward for more. And we will get back to you very soon. Well, not soon, but after some time, we will ask for some initial report, but we will keep that very simple, so not much of an effort, but we will ask you for short reports, and then we will also put some information about your initiatives on the website. We will then maybe, if you can give us some nice photographs or whatever, but we will contact you in this way. So we will also help you to become more popular in the scientific community. So thank you very much to the speakers, to the organizers, to the audience, and goodbye.